Okay, let's start. So thanks everyone for attending this webinar on phenotyping data management. So we are quite people from URGI organizing this webinar. There is Vanita Oreram, which is data steward hired by the Provide project. There is Cyril Pommier, who is the leader of the Data Federation Services at URGI. He is also the co-leader of Elixir Biodiversity, Food Security and Pathogen Science Tier and Elixir Plant Science Community. And he is also a member of the steering committee of MyAPI and Crop Ontology. There is Nicolas Francillon, who is Senior Data Manager at URGI, and is also the Data Manager co-WP leader of the MetaServe project. And myself, Michael Allo, I am the Deputy Director of URGI and the Data Manager Task Leader of the ProWild project. So a brief introduction. So it's just 2 slides of introducing the ProWild project. And the project is the organizer of this webinar. So the ProWild project is a European project with 18 partners from 11 countries in Europe, and it is coordinated by INRAE, France. And the duration, it started in September 24 until August 29. Just a slide to summarize the activities of ProWild. So there is 3 focus crops: wheat, sugar beet, and oilseed rape. So it is the work on the crop wild relative with in situ, ex situ, on pre-breeding level, with a lot of work on collection, preservation, characterization, and also breeding for the future. So just to introduce, there is challenges for the sister projects of the same call, Cuisine, FruitDiv, and ProWild. So the European Commission highlights the need to close cooperation between these 3 projects, and a key point is ensuring compatibility between the data generated by the 3 projects. And we find an opportunity here for the phenotyping data that we have already worked in other projects, such as agent in the frame of Elixir and emphasis, with the same bioinformatics partners such as IPK, INRAE, Wageningen University. And there is a clear benefit of agreeing on the use of the same minimal MyAPI the template to manage this phenotyping data. The agenda of this webinar will be the quick introduction I just made. You will be able to ask a question if you have. After that, you will have a plant phenotyping data management presentation by Cyril Pommier, followed by a question and answer, and then a live demo of the ProWild data management space and tools by Vanita, and then a question and answer, and to finish a conclusion. So is there any question on this short introduction? So feel free to raise your hand if you have a question on the agenda or on the ProWild project before the presentation of Cyril. No, nothing. Okay. You can also ask question in the chat if you want, and then I will let the mic to Cyril.
Thank you, Michael, and thank you for the introduction. I'm going to share my screen, which should be done now, and switch to full screen. And let me... Okay, now you should have the right screen.
Perfect.
Okay, very good. Very good, very good. So, I'm going to quickly present you plant phenotyping data management solutions, why we should do it, uh, and what are the most pragmatic and effective solutions. Everything that I'm going to present you has been used in Agent and other projects and is a result of the joint effort from a lot of international initiatives, and in particular the Elixir infrastructure for life science data and emphasis infrastructure for plant phenotyping data, plant phenotyping in general. So just I know that you mostly know that, but I want to frame very clearly the scope of plant phenotyping here because this is a highly polysemic term. And in fact, even among biologists, when talking about phenotyping, we are talking about different things. Of course, the phenotype is a result of the interaction between a genotype and its environment. And when we are talking about a trait or phenotyping variable, we are talking about something that needs a definition of an observable characteristic, such as yield, for instance. And the phenotype is the association of this trait, this variable, with a value. For instance, yield 6 tons by hectare, which might be a little bit more too much. Anyway, phenotype also correspond to different properties, morphological, physiological, biochemical. For instance, the protein concentration of grain or of whatever tissue is considered as a phenotype, or agronomic properties. Can be quantitative, so numbers, figures, or qualitative, with disease notations, or simply stating that the fruit is red, for instance. It is highly heterogeneous. There's a lot of creativity in the phenotyping community. The environment is very important and is key of defining the phenotypes, but it's not really part of the definition of primary phenotypes. In other words, of the characterization of gene bank material accessions. And finally, as I quickly said, it is a very polysemic term because for some researcher, it will be the measure of something in a phenotyping experiment. For other scientists, it will be the characterization of a plant genetic resource. For other communities, it will be association with genetic QTL-GWAS, and it's not the same level of data aggregation derivation. And it can even be an association with a gene function. Furthermore, the phenotype data life cycle that you are living is highly complex, and it begins, of course, with the data acquisition, with raw data, phenotype, and environment measurement, definition of variables, with single plants or single microplots being measured, each plant or microplot crossing several factors, the genotype, the cultural practice, etc., etc. And this raw data can be then derived, reduced to be able to obtain simplified data, such as, for instance, mean on genetic resources over several repetitions. And from these derived data, because it has been simplified enough to answer certain scientific questions, there can be a publication of knowledge such as variety charger is resistant to fusarios in their intensive cultural practices. It means that when talking about raw data, we are more talking about phenotyping and environment variables with some data cleaning and traceability, reproducibility, and provenance, so how the data has been measured and cleaned. When talking about derived data, we are more talking about indicators with new computation for each scientific question. Now, the purpose of the presentation of the talk I'm giving is about data reuse and data management. But why should you be interested in that? It's mostly around this idea that one experimental dataset, so one raw dataset, can lead to many derived, specialized data sets, many paper reviews. And for instance, there is this example of a multi-site experiment, indoors plus fields, in 2014, which led to the publication of 3 data sets dedicated more to genetic, genome-wide analysis or genomic prediction. 2 other dedicated dataset, which led to publication of physiological knowledge research result, another one on multiomics, another one on crop models, etc., etc. So one raw data, several derived dataset. And this is really the key, which means that to have interestingly reusable dataset, you must have with us a focus on the correct documentation of this raw dataset. This is something that we have put forward in that paper in Trans. Now, this being said, it's what do we all get as a community of preparing phenotyping data reviews? So now, how do we do that? So what we need to do is to document and trace trait methods and protocol, how the data has been obtained. Within each community, each project, so provide here and probably provide on Cousin and Frode, agree on variable trait methods on unit. Avoid something that I've seen a couple of times where in a final project meeting, we suddenly realize that data cannot be compared among partners. We need also, of course, and you know that, list and identify very clearly and without any ambiguity the plant materials, genetic resources used, localize the experiment, organize raw and derived data, document all data matrix columns. I've seen recently again some data matrices, so Excel files with 1 column, pH John 1 and 1000 grain weight chain 2, etc. We must be able to understand what are those columns without anybody having to call you on the phone or send you emails to understand what exactly those data mean. So those are the general objectives. How do we do that in practice? So there is a guiding principle, the FAIR data principles. I assume that a lot of you have heard about it, so sorry for the repetition. FAIR stands for Findable, Accessible, Interoperable, Reusable. It has been published in 2016. It is a core principle of a lot of open science policies in Europe. Findable means simply that your data set must have IDs, must be indexable, must have metadata description. Accessible, that simply it use open protocol on that with a few clicks you can access it on the web. Interoperable with good description with semantics and vocabularies, what I'm going to present to you right after that. And reusable, the key point for you is probably the license, which state exactly what are the conditions of data reuse. This is a very general principle, but we have to be very pragmatic when we want both computer scientists as me and biologists as you to build shareable data. So for this, there is a 5-star open data principle from Tim Berners-Lee, one of the founder of the web, that states that to be open, the data must be first, of course, on the web with a license, then in a structured format, then in a non-proprietary format, identified with URIs, so that's an implementation thing, but it can be also simply a DOI and linked to other dataset. And what we are aiming at, in fact, is simply to have your dataset at the end of ProWide available on the web with a DOI so you can be cited and you can get credit from your work and with consistent documentation. And the last step, this is our job, we will manage. This is not going to be yours. So this being said, really in practice, how are you going to do this? Of course, there are a lot of tools to be able to do that. There are information systems to collect the data. Vanita is going to present you right after what can be done with Excel, Validator, MyAPI, etc. So extension of what has been done in Agent. There are more advanced information systems designed especially for high throughput phenotyping in emphasis with Fizz or PIPA. A lot of other script workflows are Jupyter Notebook, etc. And to ensure long-term sustainable access to your results, we need data repositories, which can be Dataverse and Recherche Data Group in France, but it is open to European projects such as ProWide, and it has been used in Agent, or Zenodo, or EMBL, EBI, etc., etc. And finally, there are data portals to access all of the data. The important thing also you have to keep in mind is to enable connection among all these circles, we need a glue in the center with global data standards, MyAPI, Breeding API, crop ontology, and guidelines on training available on the RDN kit. So let's dive in those plant data standards. There are three types of data standards that we are talking about. The first one is about structure, and this one is really important for you. It's about formatting and organizing your data and your data description. So in other words, the metadata. It's MyAPI is definitely the reference for that in plant phenotyping. It allows also to encompass phenotyping variables and biological material description. And this has been built with an interdisciplinary cooperation between biologists and computer scientists. And those phenotyping variables are integrated using another type of data standard, so the semantic one, the ontologies, in other words, which are about describing the data with controlled vocabulary, simply list of term, of name, and definition of phenotyping variable, following the triplet trait method scales that I'm going to show you after. And this is mostly biologist driven because the intelligence, the knowledge here is in your head. There is also biological material which need persistent unique identifiers, so DOI, accession number, etc. And finally, I'm not going to talk too much about technical data standards, which is mostly the breeding API, because I understand this is not necessarily the most important for ProWide, at least at this stage. So to dive a little bit more in detail, MyAPI, the plant structure standard, is biologist friendly with clear definitions, clear examples. It relies a lot on Excel templates because it is easy to manipulate and because there is no need of abstraction on complex ontologies and things. And we as a community, as a MyAPI community and beyond, have made a lot of efforts on trainings. Since 2018, there are almost one training a year, and there is a possibility to build a dedicated training for ProWide. It's a minimum and sufficient list of metadata, which is both computer scientist friendly with an explicit data model, as well as validation framework and toolbox that once again Vanita will demonstrate to you. And it includes established standards, such as in particular the multi-crop passport descriptors, which is very important for the gene banks, and the crop ontology for variable. So MyAPI is a specification which has been implemented in databases and data repositories. in web services, so especially the breeding API, which is an international collaboration like MyAPI, and it's a technical specification, so probably not your main concern. There are file exchange implementation, so the MyAPI Excel templates that we are going to show you, project workflows such as the agent one, and ISATAB and AeroCrate for more advanced and technical approaches. And there are the ontologies and all implementation, but those are for ontologists, in case there are any ontologists among us today. So, speaking about those ontologies, I want to focus especially on the crop ontology, which allow to describe plant traits and characteristics. Crop ontology is in fact two things. First, it is a representation model, a framework based on a triplet stating that what you are measuring, so phenotyping or environment variable, is the composition of a trait method on scale. The scale being either the unit or a notation scale. And on top of that, the crop ontology can be, as you can see here, a collection of ontologies by species in which you can pick existing phenotyping variable. But what you've got to keep in mind is that in case in the wheat crop ontology, you don't have any phenotyping variable corresponding to your own practices in ProWide, you can totally create your own ontology specific to your project. So in detail, the crop ontology trait method scale relies on the definition of the trait, which is, for instance, here the plant height, which can be measured with several methods. So the total height, for instance, from the ground to the first tassel branch, to the last expanded leaf, or to the youngest growing leaf. And when talking about high throughput phenotyping, it can be also up from the ground to, the highest pixel corresponding to the plant. And on top of that, you have several units. It can be international system units, centimeter, millimeters, or the number of pixels. So we see that with one trait, plant height, we have 9 different variables by picking plant height, total height in centimeter, or in millimeters, which is a second variable, etc., etc. The fact that you decide to compose those phenotyping variables that way will help to make your data really comparable among the different gene banks, among the different partners, institutes of ProWide. So very quickly, those standards have been widely adopted worldwide. So it's the aim for us is to make it transparent for users that you use standards without even noticing it. BRAPI and MyAPI are two sides of the same coin. There are several databases that implement those. There are BRAPI tools. Agent has been using it extensively, and Elixir and Emphasis are using these standards as their core reference. So the MyAPI specifications, it's available on the myapi.org website, which is very small here. And there has been the latest publication in 2019, and there will be another one this year if everything will go well. The MyAPI specification is simply a table, a list of section, investigation, study, biological material, observation variable, etc., with in each section metadata fields such here, for instance, variable IDs, variable name, with definitions, examples, cardinalities. In other words, everything you need to clearly understand what you are talking about. And since this is a flat list, on top of that, we need a data model to link those different sections and concepts. And to state simply that in the investigation, you have several studies, and in those studies, you have several biological materials that have been studied and measured. And to give a very quick and short overview of what all of those sections mean, what you have to keep in mind is that the investigation is the entry point of MyHappy. It's a full data set. It can be a data set encompassing fields experiment, greenhouse experiment, no problem about that. And the data set contains data from one to many locations over 1 to many years. Hence, being able to cope with, of course, crops data over several years, but also with perennial plants such as trees, grape, and so on. And the investigation, the dataset contains several studies. The study is, in other words, an experiment. There is one study per location. You can have multi-site studies during 1 to several years, as you can see in that example. And finally, you've got all the series of measures themselves. That can be either, for instance, at the level of the plant or at the level of the microplot or pot in a greenhouse, with several assays which are the different variable defined in your own ontology. To finish, I will give you a quick example of the MyHappy template in practice using this data set published following this DOI. So the ID I've mentioned to you, in the FAIR principle introduction, which allows to make Emily work fully citable. The first thing you have to notice here is that the MyHappy template is self-documented with a README sheet that allows you to give you all the definition of the different fields that you will cross. Therefore, you don't have to run from your template to the website and so on to understand what you are dealing with. So the investigation or dataset is a very simple sheet with its ID, its title, its description. So description is really the abstract of your dataset. And the important thing here is the license that you can use. CC BY is a set of open license, Creative Commons, that protects researcher rather well and gives good opportunity of reusing the data. Then you've got your list of study with both study ideas which are short name, easier to manipulate either in Excel or in R scripts, such as guide 12, 13, etc., but not necessarily very explicit. Therefore, you need the full title, is the human readable title, description, start date, end date, etc. And a lot of other metadata. I haven't shown all of them. What you have to keep in mind is that you've got the locations here, a lot of MyHappy descriptor, such as the design, the grow facility description, etc. And MyHappy is a minimal standard, which means you can add additional information on top of it if you want, of course. If you want to define project-specific custom columns, that's totally doable. Then you've got the persons with their ideas, and the interesting thing is that there is a role allowing you to list not only the PIs for your institute, but also the person who runs the experiment, the researchers, the analysts, etc., etc. Then, one very core and key sheet is, of course, the biological material with mandatory fields such as the biological material ID and recommended fields such as the material source ID and the material source accession number. And on top of that, you can add, once again, any additional information that you see fit. In that example, for instance, the researchers have decided to add the parents of each accession. This is not part of MyAPI, but it is super useful to them, of course. Keep in mind also that there are two levels in the biological material. The material source, which is the gene bank accession number, so as you can see here, material source accession number, or a cultivar, commercial varieties such as Renan, and the biological material, which gives traceability of the seed lot. For instance, here, the 2023 seed lot for the accession B73 INRAE. This is not mandatory, and you could simply duplicate the information of the accession into the biological material. The optional variable, another mandatory sheet, is once again based on the trait method unit triplet. It's a minimal template. You see all of it here with the variable, the name, the method name, and the scale or unit. And that should fit most of your needs. In case you would want to go much further than that, there is also the crop ontology template, which is way richer, but as far as I understood, this is not something that you are going to need for ProWide, but I wanted to cite it nonetheless. Finally, the MyAPI data can be stored in a dedicated data sheet, here, for instance, or in separated files if you have several files corresponding, for instance, to the microplot level observation, to the accession or genotype level, etc., etc. The important thing is that the format of those data files is absolutely not constrained in MyAPI. As you can see here, there is this format used in the drops datasets that I've been showing to you so far. And in Agent, the data files have been organized totally differently. Of course, there are commonalities. So what you will need to do in ProWild is to agree on a common format, but most of the time it's a set of metadata columns with the accession number, the experiment IDs, parents possibly, and then the list of variables with their different values. I'm done with this very quick introduction overview. And if there are, so first I would like to thank a lot of people. And if there are any questions before Vanita dives into, in practice, how you are going to do it in ProWide, let's discuss.
Thank you, Cyril. There is a question or comment in the chat by Felipe. Quick comment on the ontology. Felipe say we should be careful about defining too many custom ontology instances, or we may end up in a situation where data cannot be easily merged or compare. And I think, yeah, it's a key point. If you want to answer, Cyril.
Totally agree about that. There is a balance between flexibility that you will need and being able to link to reference ontology. And for that, there is this variable accession number, trait accession number, etc., which contains identifiers from reference crop ontology or from the plant ontology, etc. And with that, you will have the possibility to import some existing variables or some existing traits, and possibly to adapt the method if the one available in the wheat crop ontology, which is very semi-driven, doesn't correspond to what you are currently doing in cousin, especially for wild relatives. So
yes, that's we are doing in ProWild for each species. We are discussing with the researcher and to have the trait. If we can find it already in the corresponding crop ontology, and
Mhm.
if not, we are creating a new variable.
If
Okay.
I understood well during our discussion yesterday, Michael Vanetta, I speak under your control. The idea will probably be that first we collect all the trait variable as draft as they are used in the ProWild consortium on Beyond with Cousin and 3D. And then there is a curation process, mostly by Vanita, to pick the right traits and the right variables in the ontologies if they exist.
Yes, I
Okay.
think it's a
Nice. Thank you.
good process. Do you want to say something else, Felipe?
No, on our side, we are, as you know, we have adopted the Germinate tool to actually manage the information. And for now, we are stepping aside a little bit from the ontology debate. We are first focusing on the actual data format because sometimes it's not quite easy for us um, to fulfill the requirements of the platform in terms of the spreadsheet templates that we need to use to upload the information. But it's only that data format to upload information is a little bit strict, okay? And we need to adhere to that if we want to use it. So we are in the process of trying to adapt this information. Ragma is working on the data curation analogally with Vanita, okay, to actually adapt the information provided by researchers to the platform to validate the whole process, and hopefully trying to automate. One thing we instead of ontologies, one thing we are considering is try to provide hopefully a lightweight procedure to code the data preparation tasks and trying to orchestrate the data creation pipelines. That's our contribution, probably complementary to the one you are working on.
Felipe, just at the
Yeah.
beginning, I think I quickly heard you are using Germinate.
Mm.
Yes.
Yeah, okay, very
Yeah,
good.
in Cousin, they are using Gemini to integrate the data and display the data. Yeah.
Okay.
So there is some constraints in the formats.
Yeah.
And I'm
So,
not
but
sure there is fully MyAPI compliant. Do you remember, Cyril?
it is mostly, I would
Mostly.
say it's mostly compliant with working with Rick van der Sede from Wageningen on this MIA-PE um, compliance problem.
Mm.
But as I said, it's not a complete solution because it only solves the problem of the data acquisition platform, but not the data preparation process. So we need to complement this part. So what to do in between? You provide the data, the raw data, and you comply with the templates required by the platform to upload the data. And what happens in between? It's what we are working right now.
Yeah, yeah. Yeah, as we
Okay.
already discussed, it's very important, I think, to keep the file in some kind of MyAPI format before the submitting it to the Germinate format to have both. It's very to keep both. I think it's very important.
Exactly, and since it's not quite evident for many people
Mm.
how to what to do to prepare and how to do this to accomplish this, we are trying to provide support coding the tasks for data preparation, but sometimes it's not quite straightforward because many existing platforms are only let's say applicable if you have some, like Cyril was mentioning, some computer science experience or extensive programming experience, and not quite lightweight, okay, for researchers in other areas. So that's a main problem.
Okay, thank you. Is there another comment or question? Feel free to open your mic.
Here.
Yes, Avi.
So I'm not an expert in this, but if you have data already or you plan to gather data, and if you have an Excel sheet, so you can run it on the MyAPI, I understand that correctly?
So MyAPI are a lot of different things currently. There is an example of big experiment that has been run in 2020, which is currently stored in several sheets, Excel sheets, databases all across Europe in 10 locations, nine locations. And it is then backport to the MyAPI template, the ones that I've shown, to prepare its publication. So it's a hand creation process. Some of the databases of that project, so I'm thinking about FAIRS, will be, as soon as we get a project to fund that, able to directly export MyAPI templates, but we are not there yet.
Mhm. And is it compatible with the GWAS kind of analysis and other type of software?
GWAS, yes, totally. No problem about that. Models, it's more complicated because models have very strict list of variables and it's more handled by ICASA. But there is a compatibility of mapping between ICASA and MyAPI.
Thanks.
Another question or comment, feel free to open your mic. If not, we give the mic to Vanita. Thank you very much, Cyril.
Yes, I'm gonna share my screen. So, okay, can you see my screen?
Yeah, try to full screen. Yeah, it's perfect.
Okay, nice. So, hello everyone. As Michael said earlier, my name is Vanita and I'm going to present the tools and the data management space that we have developed with a big focus on the management of phenotyping data generated by the ProValue project. So we have created an online data management space for the ProValue project. This space is based on the Nextcloud collaborative platform, and it allows all partners members to share and manage data files that are still under creation, but also to collect and validate metadata before its submission to international archives. So here you can see what the folder structures look like, but we will take a closer look at it in the next slide. So it kind of looks like a Google Drive, but it is one, which allows us to work together in a secure environment that will only be available during the project period and for project members' internal use only. So in short, this platform is our central hub for internal data management within the ProValue projects. Okay, so the structure is designed to be simple and intuitive so that everyone can easily find and upload their data. So at the top level, there is the root folder, ProValue, which is the entry point of the Nextcloud storage space. From here, you can reach separate folders for each species studied in the project. And to make the navigation easier, we have included the guidelines document that describes how data collected and produced by the ProValue project is organized. So each species folder follows the same subfolder structure so that data is organized consistently across species. So I'm going to show you one species folder, but it's the same architecture for all of them. So I'm going to go to the old seed rape folder. Within each species folder, you will find data types folders. so phenotyping data, genotyping data, and genomic data. We have also included a global README file that will document this the entire folder architecture. So where to put files, how to name them, and which metadata template to use, for example. So please always check the README before creating new folders or uploading files. So now let's move on to what's inside the phenotypic data folder. So inside, you will find a folder for each partner working on this species. So here you have INRAE and UniPA. And you will also have the MyAPI template that have been discussed earlier. And once again, a README describing the folder architecture. So that's it for the storage space. Now I'm going to explain how to access the storage space for the first time. So it's a simple 3 steps procedure. The first step is to create an account. So you can reuse a virtual identity provider called Renater, which can be used to access some services as Nextcloud. So to create an account, you must fill out an account creation request form that you can have at this link. And you only need to fill the form with the required fields that are your last name, your first name, and your email address. So here you can see that you can view the page in English if needed, because everything is in French. So step 2 is you need to initialize your Nextcloud account. So to do so, you need to log into Nextcloud using this link and search for CRU account in the drop-down menu. So you can just tap CRU on the search bar and you will find this. And once you have clicked on that, you will be asked to log in with your account credentials. And once you're logged in, you will see an empty interface, but no worries, it's normal because it's a 3 steps procedure. So the last step is to send an email to this email address, so proviledata@inrae.fr, with the email address you have used to create your account so that we can add you to the access group. You will then receive an email when your account is added and the provide folder will appear on your Nextcloud interface. So I have shown you how to access for the first time, but do not worry, you won't have to do this every time you want to access the storage space. You will just have to log into Nextcloud, search for CRU account from the drop-down menu, and you will see the provide folder. So now I'm going to speak about how you can upload files to Nextcloud. There are 2 methods depending on the size of the file you want to upload. So for large data like genome or genotyping data, it's recommended to use the desktop client. But for small files like phenotyping files, you can use the web client. So we have seen how to access and how to navigate the data management space. So I'm now going to introduce the tools that we have developed to help you. So as Cyril said earlier, phenotyping data must be submitted in MyAPI file format. And there is a tool called the MyAPI validator that can be used to check whether the MyAPI file has been filled out correctly. So in case of validation errors, it produces a biologist-friendly detailed report. And this tool is available on Nextcloud to automatically validate files that are uploaded in specific folder of the data management space. But you can also try the MyAPI validator at this link. So you just need to upload your file and it will create a report file where you can see the errors of your file. So to validate a MyAPI file in the data management space, you need to download the template, which is available in every phenotypic data folder in the data management space. You need to fill it, and then you can upload it into the to validate folder. So here. So each partner's folder contains one to validate folder. I've shown the one for the ENN for the oilseed rape, but every partner's folder have one. So once you have deposit your file in the to validate folder, the file will be validate with the MyAPI validator, and it will be moved either to the valid folder if it's correct, or to the invalid folder if it contains error. And in this case, you will have the MyAPI file, but also the reports containing the explanation of what is wrong with your file. So now we are going to see some examples of errors that you can have in the report file. So just a reminder that the MyAPI validator does not validate the data, but only the format. So here, for example, we have an error from the report file. We can see that this error is located in the investigation sheet, and it's related to a constraint not being met for the investigation title field. So here the validator has checked the values in the investigation sheet and found that the investigation title is mandatory, but has been left empty in the file. Here in this other example, we can see that the error is located in the study sheet and that it's a type error for the study start date. So here the validator has checked the values in the study sheet and found that the study start date must be filled with a date, but has only been filled with a year. So it makes an error. For this example, we can see that the error is located in the person sheet. It's a foreign key error. So a foreign key is a foreign key is a field that refers to the unique ID of another sheet, creating a link between related data. So, for example, here the study ID of the person sheet and the study ID of the study sheet. So here the study ID in the person sheet need to refer to a record in the study sheet. So the validator has checked if it matches an existing record, but it does not. So it reveals an error. For this last example, we can see that the error is located in the study sheet, and the type of error is missing columns and an unrecognized column. So the validator has checked that the field names in the study sheet were intact and found that the study title has been named. The study title name has been changed in the file. So it creates an error. You can't modify the template. So, but you can't modify names of field, but you can add some other ones. So errors such as unrecognized column should be ignored if you have added the field yourself and not modified an existing field. So now I'm going to show you how to upload your file in the correct folder So I'm going to open the next cloud. So here you can see what I have shown you in the screenshot. So for example, if I go to with phenotypic data and INRAE. So here we have the three folders, so invalid to validate and valid. And we are going to upload the file that I have prepared. So now we just need to wait 30 seconds, 1 minute for the file to be moved into another folder. So it will be moved either on the valid folder or in the invalid folder. So we can check the invalid. It's not here. It would have been great if it was. It's not in the valid either. Just need to wait.
The validator is automatically launched each minute when a new file is uploaded.
Oh, nice. It's here. So we can see that we have our file and a report. So we're going to see what's in the report. So here the first error is located in the investigation sheet, and it's a type error about the associated publication field. So we are going to see what's wrong with the file. If we go to the, oh, I'm here. In the investigation sheet, we can see that the associated publication has been filled with a DOI, but we are going to see what is written in the README for this field. So for associated publication, we should have a DOI, but in the example, we can see that there is DOI double dots and the DOI. So we are going to modify this and see if it works. I'm going to do all the errors and then we will re-upload our file. So the second error is located in the person sheets. It's a type error again about the person email field. So let's see what's inside the person sheet. Okay, so we can see that here we have an email, but for this one, we don't have one. So we are just going to transform it into an email for it to work. Okay. The third error is located in the biological material sheet. We can see that it is a unique error. It's written that this field is a unique field, but it contains a value that has been used in another row. And we have a primary key error saying that the values in the primary key fields should be unique for every row. You can see that in row 3, we have the same as in the row at position 2. So let's see. Okay, so we can see that this one is twice in the file. Every other metadata is the same. So we can say that this line has been duplicated, but we only need one. So I will delete this one to have only one. And I think that's it. We only have warnings after. Oh no, we do have another one with the observed variable sheet. So it's a constraint error. So a constraint not being met. It says that the field value does not conform to a constraint, and this field is scale name. which is none, but it is mandatory. So we will see that in the file, in the observed variable sheet. So it was a scale name missing. We can see that here the scale name is in red, so it's a mandatory field but has not been filled. So we're gonna see what the method say about this. It says that it has been expressed as a percentage, so we can just add a percentage here for the error to disappear. Okay, so now I'm going to save this file and re-upload it to the to validate folder. Okay, we'll just need to wait 1 more minute to know if our file is valid now. It's still in the to validate folder, so it has not been processed yet. Oh, nice. So we can see that our file has been moved to the valid folder, and we still have a report, but only containing warnings. But we don't really care about the warnings because those things have been taken care of by the validator. So for example, it has removed empty columns and changed the type for some entries. So everything looks good and we have validated our file. So that's it for the demonstration. And I would just like to acknowledge all the people that have been involved in the development of the myopia validator and the URGI team.
Thank you very much, Vanita. It's a really good job to have a real live demo, and it worked. So congratulations.
I'm happy.
Yeah. Uh, yeah. Feel free to open your mic and ask question on the tools or on the demo. It's your time.
Okay, a quick question here. So thank you very much. I was really happy with the presentation. It has been uh, thrilling for me because I really like these things. I think many people believe this is quite quite plain work, but it's quite important for this kind of projects to actually work and smooth and have seamless integration between things. So one quick question, Vanita. This validator, is it an external tool? Is it integrated in your data management tool from INRAE that you are adopting in ProWile?
It's an external tool that have been developed for the Agent project
Okay.
and that we are using in the data management space.
Okay. Do you know what is the technology you have used to check the problems and provide the reports?
Yes, I think it's frictionless.
Yeah, it's frictionless.
Okay, frictionless. Okay.
Yes.
Yeah, this is the framework for data validation that we have already discussed with Stefan Weiss in our work
Stefan.
package. Yeah, Stefan is. I've seen Stefan is also involved. Yeah, in Agent. Okay, so this is great. So I think it's a very good example showing that this is a way to go. At least it's a valid approach to solve this problem. And I think it's a quick question. I was just discussing with Rachma at the same time you were doing the presentation, and I think perhaps we can contribute back or collaborate to improve some things. For instance, to improve the one thing that could be useful is improving the output of the validation. So instead of having a plain TXT, which is good, it's functional, but not but it's not sorry, the most user-friendly, let's say, uh, approach for many users. We could try to create not a very complicated thing, but an HTML output using, for instance, Pandoc. So instead of producing the TXT, we produce a Markdown file that we can quickly convert to an HTML using a template, and that would be great. Also, you can embed this information in the HTML web plate in any web-based interface, actually. And I think it could be a good way to collaborate.
Yes, but the Excel validator can create an HTML report. I
Okay.
just choose to have a TXT report. But
Okay.
we
Yes,
can
good
create
to
a Markdown and an HTML too.
Okay.
Yeah, there is a standalone version, a web version. You can try it and give feedback, and perhaps we can improve also that. Yes,
Okay,
Cyril?
wonderful.
Yeah, just to complete that, so at URGI we have also the web version available that Vanita is currently showing, so that's great.
Okay.
Keep in mind that frictionless have its pros and cons, and that Matthias Bauer have tackled a lot of limitation. So yeah, collaboration with him to extend on his work would be definitely a plus if you want to add functionalities and so on.
Okay.
Knowing him, I think he would be happy.
Okay. Good to know.
Yeah,
It's good to know.
the tool is open, and yeah, it's in Git, and yeah.
Yeah, it would be great for us to try to come up with something similar for Germinate. Also to automatically validate the templates using an analogous procedure. This way we can provide another complementary solution for our tool as well. So
The Excel
thank you very
validator,
much.
the Excel validator relies on configurations. There is one configuration for agent, another for MyAPI, and you could create a Germinate configuration.
Wonderful. Okay. Well, let's talk a little bit more about that. Thank you very much. Really good presentation.
You're welcome. Another question or comments? Feel free to open your microphone. If not, I will share my screen.
Yes.
For the conclusion. It's okay, you see my screen? Yes, okay, nice. So, just a quick conclusion. So, take-home messages for the ProWild partners. So as you see, the template, the MyAPI minimal template, it's okay. The guidelines are online. The data space, the validator tools are all available to manage your phenotyping data. So no, the next step for you is just for the people who haven't done yet, create your account to the data management space. Start to fill the template, and feel free to contact us for any support at this address, this email address. We can help you to fill the template. We can also give you advice if you have some issue on the data space and so on and so on. So all the tools are available. So let's start to use them. And take a message for the other projects or for causing or fruit div. The MyAPI minimal template is available. I added the URL there. Also, the standalone version of the validator is available. You you have the URL here. And if you are interested to the data space of other tools presented, feel free to get back to us and we can start the discussion for collaboration between our ProWild and Cousin and Frondiv, and it's very important. About the MetaServe project, URGI will implement the same data space and tools dedicated to your project. So if you have some feedback and some specific needs, you will be able to discuss with Nicolas, who is in charge of this for the MetaServe project. And we also can plan a training this year. For example, I put you the link to a training we have done 2 years ago, and we can look at the training material at this address. And if you are interested for a training, we can set a training. And to finish, just the small advertisement of the next webinar. It will be a webinar about sequence data management, data submission to the EBI, both the INA of the biosamples archives. And the webinar is available for you all. The date will be the 7th of April, and you can register to this webinar on this address. And also, we will give you a link to answer a very quick survey. It's very quick. It's less than 1 minute about this webinar to help us to improve our webinar series and to have some feedback from you. Yes, I want to thank all the people who presented, Vanita and Cyril. And also, I want to thank the ProWild project who funded this webinar, and especially Jacques Legui for the coordination. And if you have some question before closing the webinar, feel free to open your microphone now.
Yeah, I do have one. We will have access to the recording and PPT, right?
We speaking. Sorry.
Oh, sorry, Gabriel. Wait, I'll open my camera. Yeah.
Okay. Yes, for the ProWild project, you can all have access to the data space. So you just have to register as explained by Vanita. You will have the slide and also the record to do the 3-step procedure. It's very easy, and then you just have to log in. And yeah, it's available for all the partners of ProWild. Another question or comment? Okay. So thank you very much for attending this webinar, and please answer the quick survey. We'll send you an email with a link after. And hope to see you for the next webinar, and also feel free to contact us by email to start collaborations on this topic. Thank you very much.
Thank you.
Thank you very much.
Bye bye.
Have a nice day.
Thank you.
Thank you.
Thank you.
Have a nice day. Bye bye.